So, continuing on what we've started last class, we're gonna now know more about meiosis. Of course, we know that we have prophase, metaphase, anaphase, and telophase. But now they're not gonna only be prophase, metaphase, anaphase, and telophase. They're gonna be one and two of them. In meiosis, it has two stages of meiosis in order to produce the haploid DNA structures of them, where we have only one set of chromosomes. So, of course, it also involves interphase, which is DNA replication, and growth, which has also three phases. Let's recap this quickly. We have G1, S phase, and G2. Okay? Then we have meiosis. For meiosis 1, prophase 1, the chromatin condenses. It was originally chromatin, like that. It is uncondensed. Then it starts to condense into a chromosome, where each one of them is made from two sister chromatids, like that. You see them? It's this way, and another one this way, and this is the centromere. Okay? So there are two sister chromatids attached by the centromere. The centriole starts to move towards the poles of the cell, the very furthest parts of this cell. Okay? The nuclear envelope breaks and nucleolus disappears. The same things that happen in mitosis. The new thing is that pairing up of homologous pairs of chromosomes occurs, which are also called bivalent. By bivalent, I mean like they are two chromosomes. Okay? They pair up together, like they're close together, then they do crossing over. Crossing over, which is also called recombination between the two non-sister chromatids of homologous pairs of chromosomes. Like this one, or let me change the color. This one and that one. Do you understand? Do you get what I mean? It's actually like this is a blue one, this is pink, so they're non-sister. That's what I mean. Okay? So crossing over of the two non-sister chromatids of the homologous pair of chromatids. Okay? And this involves swapping sections using enzymes, sections of the chromatids using enzymes by cutting and recombining, okay, parts of paternal and maternal chromatids at the chiasmata. At the chiasmata, yes. This is the part where they swap together, chiasmata. Okay? So again, this involves swapping sections of chromatids using enzymes. Like the enzymes break it down, okay, where they can be now used to cut and recombine. Like we cut the blue part here and we join it, recombine it there. We cut the pink part here and we recombine it there, okay? So there happens swapping sections of chromatids using enzymes by cutting and recombining parts of paternal and maternal chromatids at the chiasmata. And this definitely leads for variation and production of new combinations of alleles. Not only combinations, recombinations, because we're combining them for a second time. Okay, recombination of alleles. Okay, that was about prophase. So in prophase 1, let's now recap what we've said about prophase. Firstly, the chromatin condenses into chromosomes. Okay, the nuclear envelope breaks down into vesicles and the nucleolus disappears. The centrioles reach or move towards the poles of the cells. And what's new in meiosis is that the homologous pairs of chromosomes are going to pair up together. Then crossing over occurs, which involves swapping sections of chromatids using enzymes by of course, like the paternal and maternal chromatids have their chiasmata part swapped by cutting and recombining parts of the maternal and maternal using the enzymes, okay? And this of course causes variation and producing new recombinations of alleles, okay? About metaphase 1 actually. We're gonna also start with a part that has mitosis included too, like this black part, where in metaphase the centrioles reach the poles of the cells. They were actually moving towards it in prophase. Now they finally reached the poles of the cells. The spindle fibers are fully formed. Okay, they're now fully formed. And the homologous pair of chromosomes line up at the cell equator. They join here. That's what identifies metaphase going on, that they align or line up at the cell equator. And also, what's new here, the independent assortment. So we know that variation is caused by two parts, which are crossing over and independent assortment. The crossing over part occurs in prophase, okay, while the independent assortment part occurs in metaphase, especially metaphase 1. Okay, so because we're gonna discuss later on that the independent assortment and the crossing over are not gonna happen in meiosis II. It's just like the simple mitosis thing going on, okay? So we know that the spindle fibers are fully formed and they align at the cell equator where the centrioles finally reached here. After aligning, independent assortment occurs, which involves rearranging, or like the homologous pairs of chromosomes arrange themselves randomly. The paternal and maternal chromosomes arrange themselves randomly, resulting in more variation where new recombinations of alleles are produced. Like, these two are gonna pair up, okay? They were actually supposed to pair up, but now that independent assortment occurs, this one is gonna go here and this one going there. So instead of these two pairing together, these two are gonna pair together. So this creates the variation between the paternal and maternal chromosomes. Okay, so this was about metaphase 1. Let's recap them together. What happened in metaphase 1, that the centrioles finally reach the poles of the cell, the spindle fibers also fully formed, and the homologous pairs of chromosomes pair up, or actually line up at the cell equator. Independent assortment occurs, where the paternal and maternal chromosomes randomly rearrange themselves, resulting in more variation where new recombinations of alleles produced. Okay? So just simply, the centrioles are going to move here, spindle fibers reformed, or actually fully formed. These line up, independent assortment, the paternal and maternal chromosomes arrange themselves, resulting in more variation and more recombination of alleles. I hope you now kind of catched it, and you can now comprehend what I'm saying, okay? For anaphase 1, in anaphase 1, actually, like, you're gonna not only memorize it, but understand what I'm saying. Like, in anaphase 1, they're gonna move here. Like, whenever you say anaphase, you're gonna recognize, or this view looks to you. They're gonna move to the poles of the cell. But for metaphase, however, they're just here in the cell equator, okay? So in metaphase, the centromere is used to divide, actually, but now we're going to say that it doesn't divide. That's the difference between it and mitosis. The centromere doesn't divide, it's still here. And the spindle fibers shorten, pulling one of each pair of homologous pair of chromosomes to the opposite poles of the cell. Like, these two go to this left part, and these two go to the right, okay? Which is why we call it reduction division. They were at first 4, then now we're gonna have 2 cells having 2 and 2. You get me? Okay. In telophase 1, it's also like the same thing as mitosis. In telophase 1, we have like the chromosomes are gonna reach the poles of the cells, then the nucleolus reappears, the nuclear envelope reforms, the cytoplasm constricts and the cell begins to divide. We're gonna now have two separated ones. And not only that happens, but also the chromosomes uncondense and uncoil into chromatins. Like if you can remember here in prophase, the first step, the very first step we talked about was about the chromatin condenses into chromosomes. We're now getting back to the starting point, okay? Where the chromosomes are gonna uncondense and unfold themselves actually into chromatins, where we now end up by having two genetically different cells, which are haploid. Like, they are haploid. Here in each cell I can find two chromosomes. At first I had 4, so that's half of it, okay? Now let us talk more about meiosis II. Even though it's quite similar to the mitosis itself, we have to also give it more attention. For meiosis II, we have prophase II, metaphase II, anaphase II, everything is 2. Again, the chromatin coils or folds and condenses, becomes shorter and thicker. As you can see, it was like thin, not only thin, I mean smooth. Now it became thicker and shorter, okay? It coils and condenses, becomes shorter and thicker, forming chromosomes where each chromosome is made of 2 sister chromatins attached by the centromere. Okay, we know all that. The centrioles start moving towards the poles of the cell, like here. This one is right there and this one is there, okay? The centrioles start moving towards the poles of the cells, and the nuclear envelope breaks up, nucleolus disappears, new spindle fibers begin to form. Now they're new, like they weren't old and they're now formed, they're new, totally new, okay? Because the other ones were broken down in meiosis I. About metaphase II, the centrioles reach the poles of the cell, and the spindle fiber is fully formed. The chromosomes line up at the equator with the spindle fibers attached to them by centromeres at a position called kinetochore. So that if you want to know the position itself, terminology. Okay? So at metaphase I, the centrioles reach the poles of the cell, the spindle fibers are fully formed, the chromosomes line up at the cell equator with the spindle fibers attached to them by centromeres. Okay. About anaphase II, the centromere now divides. In anaphase I, we said it doesn't divide, but here we said it's just the casual mitosis, so they're just gonna divide, okay? They're not needed anymore. I'm dividing them. The centromeres divide, okay? The spindle fibers shorten, pulling the sister chromatids to the opposite poles. Last time we were talking about anaphase I, we said that the spindle fibers shorten, pulling one of each pair of homologous chromosomes. Now they only pull the sister chromatids. Okay, so this is a chromatid and this is another one. They're pulling only the chromatids to the opposite poles. About telophase, the nuclear envelope preforms, nucleolus reappears, the chromosomes reach the poles of the cell. Like they were in the middle or somewhere. They're now going to move apart because, you know, the cleavage furrow is going to form, and then you're going to have the fiber that's separating them, so they're going to separate and become 2 different cells. Okay? The chromosomes are going to uncoil to become chromatin, the cytoplasm constricts, and cells begin to divide. Then we're going to talk more about cytokenesis. Okay, this is the part of the cell cycle in which these cells are going to fully divide, producing 4 daughter cells, each with haploid number of chromosomes, which means it has only one set of chromosomes and it's haploid. Okay? Now we're recapping what happened in meiosis I and II. For meiosis I, we know it's a reduction division. Like we started with 4 or something, and then we ended up at 2 chromosomes, or actually chromatids, something like that, okay? Pairing up the homologous pair of chromosomes, which are now bivalent, leading to crossing over between non-sister chromatids of homologous pair of chromosomes, which involves swapping and the sections of chromatids at the chiasmata, the non-sister chromatids during prophase 1, okay, of which we said that it involves the enzymes when it involves the cutting and recombining of parts at the chiasmata, okay, to cause variation and new recombination of alleles. Homologous pair of chromosome line up at the metaphase plate, or actually the cell equator, like aka it's also known like cell equator, metaphase 1, showing independent assortment where homologous pair of chromosomes arrange themselves randomly at the cell equator, also causing variation. In anaphase 1, no division of the centromere, that's the difference. So the spindle fibers are going to shorten and pull one of each pair of homologous chromosomes to opposite poles of the cell. In telophase 1, the nuclear envelope reforms, followed by cytokenesis, which ends up by 2 genetically different daughter cells with half number of chromosomes. Okay? Now the summary of meiosis II is the centromere divides. That's actually not only the summary, but the differences from it and mitosis. The chromatids move toward the pole of the cell. The centromere divides. Nuclear envelope reforms, chromosome decondenses and becomes invisible as